Neuroglia is not just a connective tissue in the nervous system, but it has many important functions. In this video, I am going to tell you what are these important functions which are critical for the survival of neuron. Hey everyone, welcome back to HM Learnings. I am Harshita, the creator of HM Learnings, where students come to clear their concept and to get the study material. Make sure that you have subscribed my channel. You can also follow HM Learnings on Facebook, Instagram and Twitter. And we are going to talk about today the functions of neuroglia. So previously also I have made one video on the glia cells. In that I talked very broadly on the functions of the neuroglia, but in this video I am also going to tell you the mechanism of each of the function of the neuroglia. So in this video we are going to talk about the two important functions of the neuroglia. In the upcoming video we are going to talk about the other important functions of the neuroglia. So in the neuroglia, astrocytes actually are the ones whose functions are tend to be more important for the survival of the neuron, and that is why more functions of the astrocytes are going to be discussed. So the first function of astrocyte is the nutritive function. Nutritive function means what? It is going to provide the nutrition to the neuron. So in this diagram you can see that this is the blood, okay, and this is a capillary, this is a lumen of the blood capillary, and this is actually an astrocyte process which is being there in the blood brain extracellular fluid, and then this is the neuron. So you can see here that there are two pathways, direct pathway and the trans-astrocyte pathway. So what is happening, that the glucose with the help of a GLUT1 transporter is entering into the blood, into the brain extracellular fluid. Now glucose can enter into the two pathways. One is that the glucose can enter into the astrocyte, or it can actually pass to the neuron directly, and it enters inside the neuron via the help of the GLUT3 transporter. So the primary pathway for the nutrition of the neurons in the brain is actually this direct pathway. But what happens that there could be variations in the blood flow, or there could be variations in the activity of the neuron. So there must be some substrate buffer, so that even though there is variability in the blood supply and there is variability in the neural activity, the nutritive function needs to be present even that also. So that is being maintained by the astrocyte. So what is the astrocyte is doing, that glucose also enters inside the astrocyte, and the astrocyte can actually store the glucose in the form of the glycogen. So in the brain, the glycogen stores only in the astrocyte. So what happens is when the blood supply is varying or the neural activity is varying, in that case this glycogen will be broken down into the glucose, then it is going to be converted into the pyruvate, then lactate, and this lactate via the help of this monocarboxylate transporter enters into the ECF, brain ECF, and then with the help of the another monocarboxylate transporter, it enters inside the neuron. Now this lactate will be converted first into the pyruvate, and then it will be further metabolized to get the ATPs. Okay, so this is one of the pathway by which the astrocytes are providing the nutrition to the neuron. That is called as a trans-astrocyte pathway, and this pathway, or you can say the astrocytes, provide food or nutrition to the neuron only in the form of lactic acid. Okay, apart from this, glucose can directly enter into the astrocytes and it can be converted into the lactate. So there are two options: either astrocytes storing the glucose in the form of the glycogen, or it can convert glucose directly into the lactate and then send into the neuron. Okay, so these are the two pathways, and this is called as a substrate buffer function of the astrocyte, because it even when the blood supply of the neuron is varying, then this the concentration of the lactate in the brain ECF is regulated or is buffered by the astrocyte in order to maintain the function of the neuron. Okay, so that is the first function of the astrocyte. The second function of the astrocyte is that it actually handles, or you can say maintain the concentration of the potassium in the extracellular fluid of the brain. So how does the astrocytes does so? So this function of the astrocyte is called as a spatial buffering, and this spatial buffering is happening because of the two properties of the astrocytes. The first property of the astrocyte is that its membrane, the membrane potential of the astrocytes is more negative than the membrane potential of the neuron. So membrane potential of neuron is somewhat minus 65 millivolts, and the membrane potential of astrocyte is about minus 85 millivolts. And we know that the equilibrium potential of the potassium is minus 90 millivolts. So by this we can say that the membrane potential of the astrocyte is more negative because it is more sensitive to the potassium. So how it can be more sensitive to the potassium? It can be more sensitive to potassium only when the astrocytes have the channels for conducting the potassium, and these channels are, you can see in this diagram, there are channels which are showing there is sodium potassium ATPase pump which is pumping the potassium inside. We have sodium potassium 2 chloride co-transporter also, as well as apart from this potassium channels are also there. Apart from this, there is potassium inward rectifying channels also which are there on the membrane of the astrocyte. So these 4 channels are going to help the astrocyte to pick up the potassium from the extracellular brain ECF and make sure that the concentration of the potassium in the ECF is remain in the ceiling level, which is 10 to 12 millimole. Okay. After this, what happens? The second property of the astrocytes which help in the spatial buffering is the gap junction. So these astrocytes are connected to each other via the help of the gap junctions. Now, with the help of this gap junctions, what is happening? The potassium which has entered at this point can pass to the another astrocyte. Then from this astrocyte, it can pass to the another astrocyte. By this, what is happening? As it passes from high concentration of potassium to the low concentration, what is happening? The potassium concentration is decreasing. So it is decreasing, and finally what happens? The potassium is released back to the brain extracellular fluid. So this is called as a spatial buffering. From high concentration of the potassium, it is delivering or traveling to the concentration of the potassium, lower concentration of the potassium, and this is happening because of the astrocyte. So this is called as a spatial buffering. So by this, the astrocytes ensure that the potassium concentration surrounding the neuron is not high. And why the potassium concentration needs to be minimum, or you can say why it needs to be maintained, why there should be not high concentration of the potassium in the brain ECF? Because if the potassium concentration is more, it is going to decrease the efflux of the potassium from the neuron. As a result, what happens? The potassium accumulates inside the neuron and it can depolarize the neuron. That is, it is going to increase the excitability of the neuron, which we don't want. So by this, astrocytes help in maintaining the survival of the neuron. So with this we are at the end of this video. I have explained the 2 functions of the astrocytes, that is a nutritive function of astrocyte and the potassium buffering by the astrocytes. So if you like this video, please like, share and comment. Follow us on Facebook, Twitter and Instagram, and please subscribe to our channel. Till then, keep learning.